Hello, today we'll present our final year project, which is titled Deep Learning Based Circulating Tumor Cell Detection and Classification. And next here, yeah.
Okay.
Here is the content of our presentation. We will start with some background information. So CTCs are rare cells in the blood, which is generated by primary cancer tumors shedding some of the cells into the bloodstream. And then usually these CTCs will eventually settle into distant tissues and give rise to metastatic tumors. And this is why they are a biomarker for metastasis. And so they have great clinical significance and can be used for disease monitoring and progression. And also they have potential to be used to estimate the risk of relapse. So some features of CTCs is that it's rare, and also because they're cancer cells, they have high tumor heterogeneity, and it can be seen in the genomics and also the morphology. Next. So how this heterogeneity will affect other analysis is that first, some of the detection strategies are label dependent, which which usually assume that CTCs will overexpress certain cell surface markers uh, that can be uh, that can be targeted with antibodies. But because of the heterogeneity, not all CTCs will be expressing these surface markers. So these kind of methods have a high possibility for false negatives. And next, there are physical methods, which include such as gradient centrifugation or filtration, and also microfluidic devices. But these approaches are also reliant on trying to exploit physical differences in CTCs and CTCs and white blood cells. But because of heterogeneity, again, a lot of the CTCs physical features are overlapping with white blood cells physical features. So these kind of techniques are also not very sensitive. But unlike the other physical methods, microfluidic devices also, sometimes allow for further downstream analysis. So some of this analysis may be done with deep learning or machine learning models, which brings us to the third method, third strategy. So um, I want to talk about the machine learning and deep learning in the context of cancer cell detection. There are two applications we can focus on here. One is the image level classification, which has been demonstrated by models like YOLO V5 and RCNN to be able to distinguish CTCs from other cells from biopsy images alone. YOLO V5 was also compared with efficient dead and center net in a study, and it showed that YOLO V5 have higher MAP 50 scores. One gap in this study is that the YOLO V5 has a balance between speed and accuracy, so it often outperforms two-step models like RCNN, which are meant to be highly accurate. But there has been limited exploration of YOLO V5 in detecting and classifying species. So another application for machine learning and deep learning is for morphological analysis purposes, where single cell level classification is first required. Here, machine learning models have been explored and showed good results by incorporating algorithms for feature detection, feature extraction. Like in this one study that has built a MATLAB-based algorithm for that. But a limitation is that these machine learning based models' performances are compromised with cells that have unusual features, which is not ideal for heterogeneous cells like CTC species for classification. So for this, deep learning models often perform better as CNN-based architectures are able to automatically learn the features from raw data. As seen from the literature, uh, where EfficientNet and ResNet 50 have both shown good performance for this task, the accuracy have reached like 99%. They're also able to distinguish between different cancer cell lines, as shown by this one study. Uh, but they have, uh, but the problem is that they lack a detection pipeline. And these results also show overfitting. So next is our hypothesis for this project. We hypothesize that one, the YOLO model will be able to differentiate between different types of cancer cells from biofilm images alone based on subtle morphological differences. And also, classification models should be able to extract features of for each cell line that distinguishes them from other cell lines. And so our objectives for this project is to apply the YOLO deep learning algorithm to detect cancer cells from biofilm microscopy images. And then we want to perform cancer cell classification between cell lines and do morphological analysis using both machine learning and also deep learning models. And lastly, we want to evaluate the performance of efficient net B0 trained on single cell images to enhance classification accuracy, and all this will be done all this will be combined into a single pipeline. Next. So this project is meant to be a part of a larger project where we aim to construct a dataset by capturing CTCs from patient blood using a microfluidic device fabricated based on the design by Che et al. However, due to time constraints, we have decided to focus on refining only the deep learning pipeline. And we have also trained the deep learning models using a dataset that was constructed by using cell lines instead of CTCs. But here, the cell lines are meant to model for the CTCs from patient blood. Next.
So moving on to the methodology part, I will start by explaining the data acquisition process. To construct the dataset used in this project, we image suspended cells on glass slide in 3 channels, as shown here. There are 4 cell lines used in this project and a white blood cell. Each image includes both white blood cells and 1 cancer cell line. After the imaging process, a few steps were carried out before moving to the next part. Start by manually cleaning the data annotation part, which I will elaborate later in the next slide. Apply these following augmentations, splitting the data into 70% for training, 20% for validation, and 10% for testing. And lastly, we export the annotations and the image into YOLO v5 labeling format. For the data annotation part, we utilized AI-assisted data annotation approach, where we manually annotate around 800 images on RoboFlow software. Following that, we trained an initial YOLO v5 model and used it to help label once we got the new set of data. Subsequently, we review and correct the labels generated by the YOLO v5 model manually on RoboFlow. We then trained a new YOLO v5 model as more images became available in an iterative manner. For the cell detection stage, this slide shows an overview of the cell detection part of the framework, both training stage and deployment stage. So we trained YOLO v5 model on labeled bright field images. After fine-tuning the model, we deployed the trained model to predict bounding boxes of the unseen cancer cell images. Following that, we cropped the detected cells into single cell. For the training stage, 2 experiments were carried out by dividing classes differently. First, 2 class. All cancer cell types are grouped together in 1 class as cancer cell against white blood cells. Second, 5 class. 4 cancer cells type belong to their own classes in white blood cells, adding up to 5 classes in total. When training the model, the parameters are controlled to be the same in both experiments. We optimize number of epochs, batch size, model weight, learning rate scheduler, and other additional augmentations. Moving on to the deployment stage. After detection and cropping into single cell images, the cells would be passed to the classifier. However, not all cells are in valid shape to be passed to the classifier. Therefore, we trained another model to filter out invalid images. Mobile net architecture was chosen for the filtering task due to its lightweight architecture. Next, we'll move on to the classification part.
Okay, so for the classification task, we divided into mainly separate 2 parts. The first one will be machine learning classification based on the morphological analysis, and another one will be the deep learning classification. So for the first one, we preprocess the data by applying Gaussian blur contrast enhancement, and which will help the segment anything model to clearly identify the possible mask, and the mask will generate a possible mask into an array, and we will set the criteria to selecting mask like based on the largest area, specify the diameter that wouldn't exist the maximum of image shape, and also the circularity that will help to sort out of the invalid shape. After that, the morphological property are extracted and stored into the data frame. To process machine learning model like XGBoost, random forest, support vector machine, and multi-layer perceptron. And this will help us to see which machine learning algorithm suit the best with our data. And the train algorithm will then be passed to the deployment stage. After we getting a crop images detected from the YOLO model, the morphological property will be extracted by SAM and passed through the train model to classify each of the cancer type. And another algorithm is cancer cell classification by deep learning model. First, the image are people say with the grayscale resize and also divided into training and validation. We also like add the augmentation to increase the size of training and prevent overfitting. And we choose efficient B0 as it's widely used for ImageNet at its balance, efficiency, scalability, state-of-art performance, and has a transfer learning capabilities. And the train model are evaluated by TSNE visualization and confusion metric, along with the classification report. Additionally, we also implement the fine-tune the hyperparameters by freezing the pre-trained weight and review the top layer, improve the model performance. And in the same way, the crop image cell from yellow will be used on the trained model to predict the class of the cancer cells. For the model evaluation, we have precision at the correctness of the positive prediction. We have recall, we have intersect over union, and also like the mean average precision for uh, cell detection. And for the classification, we have the accuracy and also the TSNE visualization to see how the model can analyze high feature space from each image and to see how well cells can cell line are clustered.
For the results of data acquisition, here is some examples of the images from the dataset. Each dataset has images in three different channels. First, there's a bright field, and also there's blue channel for the white blood cells and red channel for the cancer cells. And on the right is an example of the annotated image that we have done using RoboCop. Next, we have some examples of some of the images that we have filtered out by hand. And the reasons for this is because the first two images are showing examples of two images. The one is that it has overrepresentation of necrotic and apoptotic cells, and the second one has an overrepresentation of cell debris or apoptotic bodies. The third is an example of what we have included into the dataset. So it has mostly healthy cells, but there is some cells that are early apoptotic, means that they have a low level of blebbing. So we have also annotated the blebbing cell and introduced it to the dataset to have more variety. And in the case that there is a cell that has more severe blebbing and it's surrounded by healthy cells, we will usually just not annotate the cells that is not very healthy, and we will still put it into the dataset.
After the data acquisition step, we ended up with a total number of 6,939 images for training with augmentation applied. For the training setup, we used 200 epoch, a batch size of 16, and the smallest version of the YOLO V5 model, which is YOLO V5S. Looking at the plot here, it shows the training performance for both experiments. The blue line represents the 2-class experiment, while the red dotted line represents the 5-class. In both cases, the model performed well. You can see that the loss decreased smoothly throughout the training with no signs of overfitting in either of the experiment. The mean average precision, or as you can see from the plots as MAP, increased steadily and eventually converged. Although both experiments achieved good performance, the 5-class experiment showed a higher MAP overall. This becomes clearer in this slide, where we can see from both the values in the table and the precision and recall curves. The 5-class achieved at 95.5% mean average precision, while the 2-class achieved at 90.2%. And also for the precision and recall curve as well. Well, the reasons that could contribute to this problem is likely to be class distribution, as you can see from the label distribution plots here. The 5-class setup had a more balanced distribution across classes. On the other hand, the 2-class had a severe class imbalance. As you can see, the cancer class dominated the cancer data set, while the white blood cell class was underrepresented. This kind of imbalance can cause the model to favor the majority, which is the cancer, and makes it harder for the model to accurately detect white blood cells. Therefore, it degrades the overall performance of the 2-class model. Another limitation we observed was that the model struggled with white blood cell detection. As you can see from the precision and recall curve, that the white blood cell results were below other results in both experiments. Looking at the confusion matrix, all cancer cell classes performed well, with over 95% correctly detected. However, about 42% of background regions were misclassified as white blood cells, indicating a significant confusion between white blood cells and background artifacts. Well, there are 3 possible reasons behind this poor performance. First, class imbalance. Second, white blood cells were not the primary target as it only appeared in the background. And the last one is its visual similarity to cell debris, which may have led the model to misclassify debris as white blood cells. A potential solution would be to include more images of white blood cells in the dataset, which would help to the model to learn better features and therefore improve the ability to differentiate between white blood cells and background noises. This is our mobile net filtering model. It reached 96.9% accuracy with low validation loss, even with minimal tuning. This shows a strong baseline performance for a lightweight and the secondary component. However, we did notice signs of overfitting, likely due to the small training set of just 650 images compared to other tasks we carried out in this project. Since this model acts as the free filter, we focused optimization on the mean detection and the classification models more. Still, we expect better performance with the expanded data set and the more optimizations.
And for the result of the cell machine learning cell classification, it shows the performance of the mask selection by Sam that here it has the lowest and was said to be the best number of the image of each cell line. And scikit-to-image library is used to extract the mask's properties like area, perimeter, eccentricity, and convert them into from pixel to micrometers. And the feature are loaded to different machine learning models with 4 label classes. And we have a Bayesian optimization as well to increase the accuracy up to like 3%. And for the results, it shows that the accuracy model have achieved this. Okay. And yeah, next.
Next.
And also next, yeah. And it shows that the accuracy model achieves is only 60% accuracy, and it can increase up to 63%. And from the confusion metric, there are mostly myths prediction on H2452 and TE24, meaning that some of the cells share some morphological features. But however, the classification report showed that all of the cell line from the confusion metric do not reach acceptable accuracy. We further investigate the feature importance to see how well the feature contributes distinct value among each cell line. Additionally, the plot also shows how the data from all cell lines are distributed. But however, it found that most of the features are normal distribution with no significant distinct, which is difficult for machine learning algorithm to classify cell line based on this morphology. So we decided not to include this machine learning to our development state. So further development, we think that it should be investigate on the adding more feature like the cellular structure, such as nucleus to cytoplasm ratio or gray level co-occurrence metrics to discover the unique nuclear textural pattern on the cell. And here's another result of using efficient at B0. The training dataset is around 1,700 images per dataset. And after we pre-processing, we adjust the efficient at B0 with the pre-trained weight and review top bit batch normalization and at top dropout rate. The model result shows that turning off the 60 epoch, it achieved a 96 accuracy and loss of 0.1. And here we can see like the before fine tuning and after tuning. And the prediction frequency is low compared to the scratch model or only transfer learning. It suggests the smooth learning and address overfitting. Also, the TSNE visualization show that A549 cell line are the most distinct high feature shape among cell lines. On the other hand, T24 shares overlapping high feature space with H2452 and HIA8. From the confusion metric, it shows that most of the cell mostly well predicted, only few images were wrongly predicted. Like mostly from H2452 with the 28 image are mispredicted. The possible reason might be some of the image are not properly sought out, like bare cell image unclear edges, which leads to close similarity to another cell line. So for further development, we could emphasize in increasing more dataset and sort out the invalid crop or low quality image. And also the limitation also up to the efficient model as well. As we use efficient B0, it takes the inputs as a 224 image resolution, which might limit its capabilities to capture the pattern and the features compared to a larger model or a more complex model. And also adjust on the fine tuning, the top layer might help on this as well.
To combine all the parts in this project together, we made a command line interface where the user can insert a folder of images or a single image to this program, and it will run through the models that we have trained, first by YOLO detection, second mobile net filtering, and the last model is efficient net for classification. And eventually, the program will results in will give the results whether any cells are detected and what kind of cells are detected and how many cells are detected as shown here in the demo.
To conclude, what we have done so far is to use YOLO V5 for detection, and it had a mean average precision 50 of 0.955, which proves the hypothesis that the trained YOLO V5 model will be able to distinguish cancer cells types from white blood cell images using subtle morphological cues. And then we have also used EfficientNet B0 for cell classification, and it had an accuracy of 96%, and the TSNE visualization of the results show that it shows 4 distinct clusters for the 4 cell lines, which proves the hypothesis that the trained EfficientNet model should be able to extract unique morphological features for the different cell lines that can set these cell lines apart. So with this, we can solve some of the current gaps in literature, such as the limited exploration of YOLO V5 for detection and limited morphological analysis of multiple cell classes from dry field images alone, as well as the lack of a pipeline that combines detection and classification together. And by using deep learning algorithms, we also address the machine learning problem where the performance is compromised for unusual morphological properties in CDCs. Next. So for future improvements, we may incorporate a microfluidic device for the capture of CTCs and white blood cells from patients. And then we can build another dataset to train the models to detect and classify real CTCs. And then we can also equip the microfluidic device with a high frame rate camera to image cells in motion in fluid to enhance the classification model by allowing extraction of more features such as how the CTCs behave differently in fluid compared to white blood cells. And for the impact of this project, the detection and classification model may be the detection and classification model pipeline may be used for transfer learning applications after they're trained on patient sample data by other researchers who need to detect these cells for their own work. Further improvements on future extractions may also strengthen the model more and may have use in clinical settings. And that's it for our presentation. Thank you for listening.